So Bob Kloner gave a terrific introduction on ischemic preconditioning and remote ischemic preconditioning and other forms of cardioprotection. And I'm going to shift now more towards some of the molecular mechanisms that explain that. So, are we making progress in cardiovascular disease? Well, the good news is that since 1955 or so, the actual mortality is down by 2/3. The bad news is that the population has doubled since then, and so we still have almost 800,000 cardiovascular deaths a year. On the other hand, we're doing much better than cancer. And of the cardiovascular deaths, about 48% of them are actually related to coronary artery disease and ischemic heart disease. So, as you heard this morning, with ischemia reperfusion, you know, the classic two factors that have been studied the most and thought to be the most injurious are both the calcium release and inability to control calcium and the reactive oxygen species burst that occurs when you reperfuse the heart. And the most powerful way to protect against this was identified in the 1980s first by Chuck Murray, who showed that if you preceded the prolonged ischemia with short, in this case 3-5 minute or so bursts of ischemia, you could actually get this very dramatic reduction in the infarct size. And subsequently, it was shown that there were a lot of pharmacological agents that could reproduce this if you treated before the ischemia. And then in early 2000s, post-conditioning, where you give very brief episodes, usually about 5 seconds, off and on during the early reperfusion period, and you got similar kind of protective measures. So what's thought to actually be causing the final injury and death to the myocyte? Well, most of the field is settled on the mitochondrial permeability transition pores as being the main culprit, and these are ROS and calcium sensitive as well as pH sensitive pores that form in the inner mitochondrial membrane, which is normally highly impermeant to any ions because it depends on maintaining a minus 180 millivolt membrane potential. And this pore has a molecular cutoff of about 1500, which means as soon as it opens, the mitochondria is depolarized. Mitochondria don't care so much about making ATP. What they really want to do is pump protons. And so if there's a short circuit and they can't control the protons, they will actually reverse ATP synthase and start consuming ATP in order to pump protons out of the matrix. So you actually end up consuming ATP rather than producing it. Also, if there's any calcium that's been accumulated in the matrix, because of that negative membrane potential, it's released and contributes to the calcium overload. And so these are called long-lasting, large, high-conductance PTP openings. They also, because of the high oncotic pressure, lead to swelling, and the outer membrane will actually rupture, and you'll have irreversible damage. But they also release cytochrome C to trigger apoptosis, or if you damage enough mitochondria and have no energy production, necrosis. Now, consistent with the hormetic hypothesis, these pores aren't all bad, and actually mounting evidence that they actually can open in this low conductance transient mode that serves as a calcium release mechanism for the mitochondria, and also generates small amounts of ROS, called signaling ROS, or sometimes called superoxide flashes. So here's what's kind of fascinating about ROS and cardioprotection. If you make a prolonged ischemic episode, you get this massive ROS burst at the end that's associated with a lot of injury. If you give preconditioning, you get a small burst of ROS during the preconditioning, and somehow that dramatically decreases the ROS burst after the prolonged ischemic, and the same thing is true for post-conditioning. So if you give a reactive oxygen scavengers during the reperfusion period, you can reduce infarct size. If you give cyclosporin A, which is an inhibitor of the permeability transition pore, you can also reduce infarct size. But what's fascinating is if you give ROS preconditioning or the post-conditioning, now you lose the protective effects. These are called the signaling ROS that activate cardioprotective pathways. And if you don't allow them to work, you get back the big ROS and you get back a lot of damage. And it turns out that if you give cyclosporin here to block the transient PTP openings that generate signaling ROS, you also block the cardioprotection. So there's this fascinating interplay between ROS and cardioprotection. And here's kind of one of the current schemas of, the cardioprotective signaling pathway that occurs that's initiated by either ischemic preconditioning or postconditioning. You have these G protein coupled receptors in the membrane, and any number of their ligands that activate them can signal. And one of the main targets are the mitochondria, where they activate mitochondrial ATP-sensitive potassium channels, or perhaps transient PTP openings to generate ROS from the mitochondria. Those ROS from the mitochondria then activate a PKC epsilon isoform, which is important for then activating what's called the risk pathway, reperfusion injury survival kinases. And these somehow basically prevent these long-lasting PTP openings from occurring and killing the cell. Adenosine ischemic preconditioning episodes has a more direct pathway connection to the risk through adenosine A2B receptors and can activate this pathway. And in addition, the risk pathway and the transactivates what's called the safe pathway, which can also be independently activated by TNF and signals through STAT3. And so both of these pathways then somehow protect the heart. So most of the paradigms that have trying to figure out how these signaling pathways actually inhibit PTP opening have focused on the permeability transition pore itself. And cyclophilin D is a major component of this pore. Pore can't form without it, and it needs to be phosphorylated, and it's thought that perhaps glycogen synthase kinase or the STAT3 pathways interfere with phosphorylation of cyclophilin D, which may be preventing the pore from opening. But I think we could also go back to the schema, and given what I've shown you about how signaling ROS prevent this massive ROS burst, wonder whether the effects of these signaling pathways is not only on direct inhibition of the PTP, but maybe somehow they signal and reduce this big ROS burst as well. And so does cardioprotective signaling work by suppressing the permeability transition towards cell death by decreasing the massive ROS bursts, which indirectly inhibits PTP opening, or does it directly affect the PTP in a way that makes them intrinsically more resistant to ROS or other inciting factors? So if we look at what causes mitochondria to generate ROS, what are the sources? Well, these are the main hypotheses for the signaling ROS: activation of these mitochondrial ATP-sensitive potassium channels, as I mentioned, transient PTP opening. This succinate-driven reverse electron transport is actually thought to be more important in damaging ROS. And what I want to talk to you today about, though, is glutamate dehydrogenase, which it turns out we think may be what's activating the source of these signaling ROS. So how do these components like dehydrogenases, electron transport complexes, and reductases in the mitochondria actually generate ROS? Well, ROS are generated when you have an electron that is donated by a substrate, in this case, let's say pyruvate, if this were pyruvate dehydrogenase. Pyruvate's oxidized, its electron is transferred to a redox site, and normally what happens is NAD is an electron acceptor and rapidly accepts it and is converted to NADH or NADPH, depending on the enzyme, right? And what happens is if you don't have NAD here, this electron sits on this redox site and gives an opportunity for oxygen to snag it and produce superoxide. So when you don't have enough electron acceptors, that's when you get ROS. The same thing is true for electron transport complex. normally, they take the NADH and they oxidize that, and its electron sits on the redox site of the electron transport complexes, and it also then is grabbed by an electron acceptor, and everything is happy. But if you don't have enough of those acceptors, then oxygen can grab it. So that's the basic mechanism of ROS formation by these different enzymes in the mitochondria. So if we look at the actual mitochondria, one source of ROS production is if you have a lot of succinate, you can actually drive electrons back onto complex 1, so they sit on that site, and oxygen grabs them and produces ROS. More commonly, you need to inhibit or damage the complexes, so you can use rotenone, so this electron, once it arrives at this site, can't be transferred down the electron chain. And similar complex 3 inhibitors or complex 4 inhibitors, like this would be like antimycin, this would be like cyanide, they back up the electron flow, so those electrons sit there and oxygen can grab them. Similarly, if you have dehydrogenases, they can act as a source if there's a limited amount of NAD to accept the electrons. And reductases normally make ROS scavengers, so they take NADPH and basically transfer the electron to a reducing substance like glutathione or thioredoxin. And if you don't have enough of the acceptors, they can produce ROS instead. Finally, there's some evidence that there may be some NADPH oxidase in mitochondria, but it's pretty controversial. So the bottom line here is that the NADH-NAD ratio is very important for producing ROS. And if this ratio is normally very low, it does become high, greater than 1, during ischemia because you can't, you don't have oxygen to use up the NADH, but there's no oxygen present, and so you can't really generate ROS under those conditions. When you reoxygenate, unless prolonged ischemias damage electron transport complexes to impede electron flow, the NADH ratio very quickly decreases. So there's only a very brief time window during which you have both high NADH and oxygen present to generate ROS. And so the question is this window long enough to generate sufficient ROS to activate these cardioprotective signaling pathways, the risk and safe pathways? So Pavel Korge, who's our mitochondrial expert, began looking at different enzymes and asking the question, well, maybe there's another matrix enzyme there that does have the right properties. What kind of properties would you need? Well, you don't want it to be active during normoxia, so perhaps you need it to be activated during the brief anoxia or ischemia. Perhaps the fall in ATP-ADP ratio could be a way to do that. It needs then to transiently generate ROS during reoxygenation despite a low NADH-NAD ratio, and then it needs to inactivate again once this ratio is recovered. And the ROS must be generated must be sufficient to activate the pathway. So it turns out glutamate dehydrogenase is an enzyme in the matrix. It's also in the cytoplasm, but that has these properties. And normally this is mainly expressed in liver, kidney, brain, and pancreas. It generates ammonia for the urea cycle, but it also generates alpha-ketoglutarate for the TCA cycle, and as I mentioned, has a low activity in heart. But if you look at this enzyme, it has interesting features. The classic dehydrogenases, like pyruvate dehydrogenase or alpha-ketoglutarate dehydrogenase, have this single redox site. So they take the substrate, they oxidize it, the electron sits here, and if there's no NAD to accept the electron, it can go to superoxide. So you need a very high NADH-NAD ratio in order to get ROS here. GDH, on the other hand, has two redox sites. And the first one is very similar here. It takes glutamate, the electron, and generates NADH or NADPH. But then it has this second site. And so the NADH generated here, then, if the enzyme is activated, binds to this second site and it sits there, and oxygen can grab it and produce superoxide or hydrogen peroxide. So if you take isolated purified PDH, for example, what you see is whether you have pyruvate present or not, if you just give it NADH with no NAD present, you get a robust hydrogen peroxide production. But once you give NAD to reverse that ratio, it shuts it off, right? But look at purified GDH. Here you give just NAD, no NADH at all, glutamate, and you get robust oxygen production. And here you shut it off with the products of the reaction. And you can show in cardiac myocytes the same thing, cardiac mitochondria the same thing. Here's permeabilized mitochondria. You see you give NADH, glutamate, and what I forgot to mention here is that the glutamate dehydrogenase is inhibited by ATP or GTP. And so in this case, if you have GTP present and you give the substrates, you don't see much hydrogen peroxide production. But as soon as you give ADP to change that ratio, you get, this is NADPH production, and this is hydrogen peroxide production. And here with ATP, the same thing. You give the substrates NAD and glutamate, and here you give hexokinase and glucose to consume the ATP and convert it to ATP, and you see this robust change. And so here, actually, in the mitochondria, you can see the same thing where you give permeabilized mitochondria, you give glutamate, NAD, and you see they produce ROS, and when you give ADP, it stimulates it further. So we figured, well, let's try this, take a long shot here, and make a mouse that doesn't have any mitochondrial GDH in its cardiac mitochondria. And so we basically took a floxed GLUT1 knockout mouse. GLUT1 codes for glutamate dehydrogenase, and we crossed it with a cardiac Cre-expressing, and you can see that we eliminated the glutamate dehydrogenase from the mouse mitochondria. And we showed in these mitochondria, when we gave them the glutamate and ADP, which in wild-type mitochondria gave a robust response, basically they did nothing. And then we went on to subject them to ischemic preconditioning protocols. Here's control ischemia for 25 minutes. Here's ischemic preconditioning, followed by ischemia for 25 minutes. This is hydrogen peroxide-induced preconditioning, followed by ischemia. And here we did post-conditioning. And here are the results. This is both looking at the functional recovery and the infarct size down here. And I'm going to focus on the infarct size. So you see, if you take a wild-type mouse and you make it ischemic, this is in a Langendorf setup, global ischemia for 25 minutes. You see we get about a 75% infarct size. That if we precondition it with ischemic preconditioning, we reduce the infarct size to about 25%, right? If we, instead of ischemic preconditioning, give it a very low dose of hydrogen peroxide, this is 1 or 2 micromolar, we get a significant protection against a reduction in infarct size. And post-conditioning, unlike Bob's experiments, they worked in our mice, and we got a nice protective effect from ischemic post-conditioning. This was just 5-second on, 5-second off, 3 episodes. And what's interesting is in the knockout mouse, here we have a similar infarct size, and ischemic preconditioning does not work. But interestingly, if instead of, in these mice, we think we need the glutamate dehydrogenase to produce the ROS to trigger the signaling. So in these mice, we would expect then perhaps that if that was correct, if we gave exogenous hydrogen peroxide, we would get cardioprotection, and what you see is a nice protective effect. And interestingly enough, these mice are post-conditioned normally. And you would that's not unexpected, because there are a lot of other things that can generate ROS after a prolonged ischemic period, because you have damage to the electron complexes, et cetera. So we looked at the signaling, and what we found was that here's in the wild-type mice. These are 4 different markers. The first 3 are for the risk pathway, and STAT, phosphorylated STAT, is for the safe pathway. And what you see is relative to control ischemic periods, preconditioning with ischemic PC activated all of these markers, so indicated both the risk and safe pathways were activated. And hydrogen peroxide also activated the pathways. In the knockout mouse, however, we see that ischemic preconditioning did not activate significantly either pathway, whereas hydrogen peroxide preconditioning did. We then collaborated with Linda Kai's group at UCLA and looked at, tried to measure reactive oxygen species with electron spin resonance. And here you see in the wild type mice, this is control level of superoxide, and when you give ischemic preconditioning, normally you see an increase after reperfusion. After 25 minutes without ischemic preconditioning, you get a larger increase. So this would be the signaling burst, this would be the large damaging burst. And you see if you give ischemic preconditioning before the long ischemia, you see a great attenuation in the burst here. In the glued knockout mice, we see actually no increase in the ROS during ischemic preconditioning. And we see a significant increase after ischemia reperfusion. And we expected to see that with IPC before that, we wouldn't actually see a reduction, but in fact, we do see a reduction. So in conclusion, what we found is that glutamate dehydrogenase is required for cardioprotection by ischemic preconditioning in mouse hearts. The finding that exogenous ROS protect GLUD1 knockout mice suggests that GDH may be a critical source of ROS, triggering cardioprotective signaling via the risk and safe pathway activation, and has these properties. It's inactive during anoxia. It's activated by during brief hypoxia by the fall in the ATP-ADP ratio, generates ROS transiently during reoxygenation despite a low NADH-NAD ratio, and then inactivates again after the ATP levels return to normal. And GDH is not required for cardioprotection by ischemic post-conditioning in mouse hearts, which, as I mentioned, we have other sources to explain the ROS generation under those conditions. So we think this could actually be what's causing ROS when transient PTP openings occur, because the transient PTP opening depolarizes the mitochondria, they start consuming ATP, the ADP goes up, it's going to activate this, and we would expect if there's glutamate present, as there is in the mitochondria, we would perhaps see it could actually explain the mechanism of ROS production by transient PTP openings, which is still pretty controversial. We're also asking the question, could they explain why ROS production is activated when mito-KTP channels open during ischemic preconditioning? And this has been attributed to matrix alkalization, and we think one possibility that we're testing is that the matrix alkylation causes transient PTP opening, which then causes the depolarization that activates GDH. So these are still studies in progress. And finally, if our hypothesis is correct, then we would expect that things that activate, that depend on mitochondrial ATP ROS production, like the various G protein coupled receptors like diazoxide or nitric oxide or calcium to activate PTP opening, all of these we would expect to be blocked or ineffective in the knockout mouse, whereas things that work distal to that pathway, like phorbol esters or adenosine, we would expect actually to be protective in the knockout mouse. So we're studying those right now. And so as kind of a summary, preconditioning here, whether it's ischemic preconditioning, whether it's elevated calcium, or whether it's safe pathway activation, we think these act through this pathway, activating ROS production, which creates the signaling ROS and then activate the risk and safe pathways, and that these may target the PTP components directly to increase their resistance to opening, We thought originally that maybe they also are responsible for the decrease in ROS, but that doesn't seem to be the case. So Ed, I might want to propose Paracelsus is the father of hormetics, maybe. This is a quote from him in the 1500s. All things are poison, and nothing is without poison. The right dose differentiates a poison from a remedy. And I want to then thank my collaborators. Pavo is our mitochondrial expert who did all the isolated mitochondrial work. Guillaume Calmette, a very talented French postdoc of mine who's also a 100-mile trail racing champion, ultra marathoner. And he did all the work with the mice, and Scott made the mice, and Linda Kai and her group did the electron spin resonance experiments in collaboration with us. So thank you very much. Yeah, I think that's an interesting question. I'm not sure how much glutamate dehydrogenase, but I would guess that there's so many scavengers for hydrogen peroxide as it goes through the cytoplasm in terms of, glutathione reductase, and catalases, et cetera, that I would doubt it could get that far. Bob? Well, as we discussed briefly on the elevator, nothing really, despite how amazingly protective this phenomenon is, the clinical results have been extremely disappointing with it. And my own suspicion is that most patients coming in with an infarct are already preconditioned. The way people have infarcts is they develop a ruptured plaque. That plaque then forms clots on it. Clots, platelet aggregates form and break loose, and clots may form and break loose. And my suspicion is that those actually induce preconditioning in a lot of those patients. So we're not really, even with post-conditioning so I'm very impressed that the remote ischemic preconditioning seems to work under those conditions and has shown positive effects. But that would be my best guess anyway.